Hello everyone, this is going to be the unboxing style video for the NA plates for our 2421 environmental isolation. A few things going forward that you're going to just have to trust me on. You're going to have to trust me that over to my left here I have a flame going. You're going to have to trust that I'm doing proper technique and that my tools are all going to be sterilized. I say you have to trust me because I'm not going to be showing you exactly how to do this. That is part of the aseptic technique video. You should know how to do that by now. So we're going to start with our 10 to the minus 4 plates. I'm going to start with this one since this one looks more useful. Now, I'm going to start by removing away the parafilm. Remember, we did put parafilm on here to ensure a nice sealed environment. Like that. There we go. Now we do have some colony forming units here. Remove the parafilm. Now I am going to be poking around. Typically when you do this, you wouldn't be doing it on the bench like I am, you'd be doing it in a BSC. However, due to us not being able to stick this camera in the BSC, I'm going to be very, very cautious. I'm not breathing anywhere near this NA plate. Remember, we don't exactly know what these bacteria are. So as you can see, we have some CFUs here. I'm going to start with a needle so I can poke and point them out to you. We have one here, 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 here. There are also several smaller CFUs here that overall I would say are probably around 70 or 80 smaller colony forming units with two larger ones. Let's take it. They are hard. They have a hard shell texture, but they are not what we would consider mucoid or butyrus. They're just a typical bacteria. Based on the texture is similar to the bacillus species that we work with. This might be a bacillus species, it might not. We haven't done any type of testing on them. But that's what we find here. Now remember, this is nutrient agar, so we don't exactly have a exact knowledge of what's going to be here. Nutrient agar is at its core a very broad agar, and a lot of things grow on nutrient agar. So identifying specifically what is here is going to be difficult without doing gram stains, biochemical testing, and the like. Let's compare that to the second plate. This is our replicate, also 10 to the minus 4. And as you can see here, there's a very large colony with another colony growing up onto it. I'm going to pull out my needle and we're going to poke around and show you a few things. Okay, let's take a look here. You have one very large colony right here, a second one right here. You have a very small one right there that's growing on top of this colony. Colonies can grow on top of each other. You also have one here, here, a bunch of smaller ones right there, 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 and there. When looking at this, a rough estimate, I'd say it's probably going to be around 20 or so small colonies in addition to the big ones that you can see. When you're analyzing these, you want to do as best as you can, looking at these and seeing what colonies are actually there. Same as before, we're going to take our loop and we are going to test it. Yeah, that is definitely a harder texture. It's dry. Actually, it feels waxy as well. That's interesting. I might take this and do some testing on this and see what it actually is just for fun. And the edges are a bit mucoid, but the intersection is definitely a drier bacteria. let's go off to the side here. Let's use this. Yeah, that is a nice buttress texture right there. And this one, that one's also buttress. You will have pictures of these on OneDrive after I'm done going through the plates with you, so keep that in mind as well. You are to use this and the individual pictures. Keep in mind that you do need to calculate the colony forming units. We want you to look at them and count the units yourself, not to use my rough estimations. My rough estimations are just that, rough simply from looking at them. Flip these upside down, off to the side. Let's pull out our 10 to the minus 5 plates. And we're going to start with this one as well. There we go. Right like that. Now this one, there's very few. I can see maybe 4 at first glance. 1, 2, 3, 1 down there. Oh, hello. That one's actually right on top. As you can see, I can move it with the needle. That's cool. That has a nice hard outer shell. I don't think it's an actinomyces. Typically, those have a hard time growing on this auger because the bacteria outcompete them. Yeah, that one's dry as well. And I don't want to go too deep into the auger without, well, damaging it. So that's cool. That right there is a cool little bacteria colony. Pull out a clean tool. Ooh, okay, that one smells. This plate actually has a scent to it. What does that smell like? It's hard to describe that smell. It's a bit acrid, which is odd. But let's take a look. Just by looking at this colony here and this colony here, this big one, they are both butchery. Oh yeah, look at that. Just like butter. So these are 2 butcherous colonies. That's interesting. I have no clue what they are. I might do a few tests on these just because that's so cool. Let's pull out my other tool here and see what we have. You can see this one here came all the way around. There is more over here, and this is a different texture. That looks more mucoid to me compared to the butyrous texture of this and that. And that smell is interesting too. It's not what I would have expected, but it's certainly a nice descriptor. Maybe someday they will invent smell-o-vision, and you'll be able to smell it as well. That's an odd smell, and I've never really smelt that before. Hard to describe. 10 to the minus 6. This one. Alright, this is a dud. No, there's bacteria in there. I can still smell. That might be the parafilm, actually. I'll have to do investigating on that smell, because that's an odd smell. That might be the parafilm, because the parafilm was in a hot incubator. But there might be some bacteria. As I'm rotating this, there is a very small amount of bacteria on the surface. Not enough for us to really investigate, but something to make note of. This one, there's definitely a bacterium. Let's take a look at a bacterial colony. This right here is a colony. And so is this. Now, there's not much here, which I find odd, but sometimes serial dilutions, it's not exactly perfect. Sometimes vortexing is not perfect. Yeah, those are really the only two colony forming units I can see. Interesting. Let's disinfect our needle. Keep in mind that on things like the last two plates, when you're calculating your CFUs, too few to count is an option. And as you'll see here, it might be that sometimes a higher dilution has more colonies than a lesser one. And that's just the nature of us doing these dilutions. It's the nature of the soil that we use and the like. I don't exactly know where this soil came from. In the past, we've had soil come from farms, and that typically has a lot of bacteria if it's a nice loam. This looks like it may have come from some sand, more of a sandy base rather than a farming base, which might explain the difference in bacteria. Because remember, different soil types have different bacteria that grow in them. Let's peel this away. And take a look at what we have here. That's quite an obvious one. Touching it, definitely Butreus. Definitely buttery. And that's it. It's the only one I see on this plate. And our last one. This is our last Na plate. This is again a 10 to the minus 7 dilution. That same odd smell again, which makes me think it's probably coming from this large bacteria here. So there is this, which is sort of a milky color, like milk, and then there's this almost eggshell-like color. This is that same buttery-like textured bacteria that we've been seeing in the other plates. This one is slightly different. It doesn't have as buttery of a texture. It has more stickiness to it, which is very interesting. We also have a colony here, I think, that could be of use. And there's more over here that are actually inside the bubbles. Remember, we did free pour these, so the bubbles might come up. That's also very interesting. As I mentioned, I will have photos available to you. Use the photos and this video to help out with your analysis of these plates.